I became interested in the possible anti-diabetic effects of Moringa in Ghana at the second Moringa Congress, and not actually as a result of the formal proceedings, but we had a number of breakout sessions where we sat down with physicians who were treating patients, and they were queried about, what do you see? You know, what do you think Moringa does? And the thing that kept on coming up, I don't know if you remember that one, was diabetes. And we had doctors, mostly Ghanaian and Nigerian doctors, who were saying, for example, that their patients were on anti-diabetic medication, couldn't afford it, and they tried them on Moringa, and it worked, and helped them. So there were a lot of testimonials. I'm trained to disregard testimonials, but I'm a human being, I can't. So you think about these and you wonder, well, has someone done the research in a more methodical way? And it appears that there are, in fact, at least a few studies which are confirming that there may well be an anti-diabetic effect in people. There are a lot of studies in rats showing an anti-diabetic effect of Moringa. But there's not time to talk about them, and they're not as fun to talk to your customers about either. Venetia Nambiar, who was also at that meeting in Ghana in 2006, published a paper fairly recently. This is work that she did quite a while ago, in which she had 16 or 17 subjects in a control arm, 16 or 17 subjects treated with dehydrated Moringa tablets, Moringa leaf powder. These are hyperlipidemics. They have high serum cholesterol or high serum triglycerides. And they were on this intervention. This is 8 tablets a day, 2 different times a day. The first time was in the presence of the investigator. The second time they were trusted to take their tablets. And there were blood draws and some comparisons of anthropometrics. Again, 50 days. And what they found was statistically significant, not earth-shattering, but the numbers moved. So the total cholesterol moved slightly from pre to post-test in the experimental, not so much in the control. Non-HDL cholesterol, the same thing, and the LDL-HDL ratio moved. Again, these are very small, small differences and small numbers of subjects, but it's suggestive that we may want to look more carefully at this sort of indication. Another diabetes study that was done reported in 2011, which I found convincing. 30 subjects on Moringa, 30 subjects, no intervention. They took 2 tablets a day of Moringa leaf powder, dehydrated leaves, one after breakfast, one after dinner. These were sedentary, hypoglycemic, type 2 diabetes subjects. They were on oral sulfonylurea, so they were all on treatment. They were certainly not overweight. They were middle-aged, and they were on a standardized calorie-restricted diet. And these numbers, I think, are a little bit more impressive. I found them to be a little bit more impressive. There's a problem with this study in that the controls and the treated weren't matched at the beginning of the study very well. And this was pointed out by others who have reviewed this study. However, the experimental group that was given Moringa started out with a postprandial, after eating, blood glucose of 200 and 30, 60, 90 days. It went down steadily and apparently significantly. So again, I would love to be able to, and part of the reason this study didn't make bigger headlines, I guess, is that there was an issue with the matching. So what I mean by that is the experimental group and the control group weren't randomized well enough so that their numbers at the beginning were exactly the same. And so unfortunately we can't blow a horn about this study and say, wow, this proves that there's an anti-diabetic effect, but it adds to the evidence. Again, a hypoglycemic effect. Small numbers of people were given Moringa or as a direct or no intervention for 40 days. These were non-insulin dependent diabetics. They were given 8 grams a day of Moringa leaves. And it was a 40-day study. And there's a lot of data on this. Obviously, the point here, the only point I wish to make is that those on Moringa compared to the controls absolutely did appear to improve in their blood glucose status. So all of this is, I think, encouraging. I can again send you the papers and we can talk more about them. As I mentioned, these studies and others were reviewed very recently in a very decent journal. This author comes to, I think, similar conclusions to what I've just told. I got another structure here, but I have to because this is I thought, a really interesting paper. Don't pay any attention to the structure, but please do pay attention to what happened. There's a compound in Moringa. It's an indole alkaloid. Chemists know all about these sorts of compounds. Structure doesn't matter, but this indole alkaloid, isolated from leaves, had a quite dramatic effect on heart injury, cardiac injury in a rat model. I apologize, I had to throw this rat study in because I think it bears repeating in a more clinical setting. What they found was this molecule, according to them, appeared to involve prevention of the disruption of the cardiac myofibrils, the fibers in the heart muscle, possibly through a reduction of oxidative stress, leading to improved cardiac contractile function. They do this with a compound called isoproterenol. It's a nasty compound that induces myocardial infarction. And so they're claiming it's the free radical scavenging properties of this molecule. I'm not sure that's the case, but it doesn't really matter. If it works, it's of interest. Just to help you interpret this, they're showing the staining of heart muscles in a normal heart. This means that there's full blood flow, no infarction. In the drug-induced heart attack, if you will, rats, you see much lighter staining and an area of non-staining. And then in the hearts that are treated with the Moringa compound alone looks pretty much like the control. And then if you treat with the Moringa compound and the artificial heart attack drug, if you will, you have, I think, quite a dramatic effect. Obviously, it doesn't look exactly like the control, and they're just showing it graphically here, percent infarction. So again, I went through this study. I read it carefully. I think it's quite a well-done study, and I hope someone soon follows up on this sort of work. They also looked at changes in protective enzymes, changes in glutathione, and so on and so forth. And they found, as you might expect, increases in these protective enzymes. They also looked at electrocardiograms and found that the rats that had been treated with Moringa compound had a more normal EKG. The same sort of work, no, I'm sorry, it's another rat study, but the same sort of work has actually been done fairly recently with brain ischemia. And again, this has to do with oxidative stress. It has to do with reduction of blood flow, in this case to the brain. I'm going to skip over this quickly, but essentially they're showing the same kind of thing, that they can reduce the damage caused by cutting off the blood flow and then allowing it to start up again in an animal's model. I mentioned that there were 2 clinical studies that were highlighted on my scientific paper search. This was one of them. Frankly, some of you may find it interesting. I didn't think it was a particularly well-done study, but I report it to you because it's part of the scientific literature. It's very recent. And in this, they're saying that Moringa oleifera application of it reduces undesirable sebum secretion by the skin, oiliness of the skin. If this is something that your customers are interested in, then this may be of interest. It's a small study. It was not done particularly well, but again, you have to listen to these, and if time and again you see small, maybe not so good studies like this, then maybe sooner or later someone will do a better study. And the final, I think this is the final study I wanted to share with you on clinical effects of Moringa, blew my mind. I was very surprised to see it. It's been around for a few years, but this is a study done in India on the anti-asthmatic effects of Moringa. And just to cut to the chase, I won't show you any graphs. They used finely powdered seeds. So they used Moringa seeds, 3 grams a day for 3 weeks. It was an open label, non-competitive study. That just means there wasn't a control. They had 20 patients, male and female, with mild to moderate bronchial asthma. They were visiting an outpatient clinic in Ahmedabad in India. And look at these results. Lung volume, this is all in the Moringa treated people. Lung volume, a measure of how much air you take in. I'm using acronyms here. Forced vital capacity is what the asthmologists or asthmatic doctors use, but 33% increase in lung capacity, 30% increase in ability to blow out forced expiratory volume, 32% increase in peak expiratory flow rate, 20% increase in forced expiratory flow. These are all indexes of how well you breathe and how strong your lungs are. And so there are increases in all these. Plus, there were quite dramatic reductions in the scores rating a variety of symptoms that patients discuss with their doctors when they go in complaining of asthma. So they rate on a 1 to 5 or 1 to 10 scale, and so wheezing, chest tightness, cough, etc., all reduced. So anyway, this is I wasn't aware of this study, or if I was, I had forgotten quickly. So it was a pleasure to find this. Let me read just a couple of things because I don't want to miss making these points. Again, I'll turn my back partly to you, but I think when we talk about the strength of the medicinal effects of Moringa, we, meaning the community of Moringa lovers, are hurting ourselves if we let, I said this, and this was a slide that I meant to put at the beginning, if we let the unscrupulous behavior of some people and companies that make outlandish claims about Moringa taint what clearly is an extraordinary plant and what truly does have great medicinal and nutritional potential if used appropriately. What does appropriately mean? I think appropriately means consuming modest amounts of the leaves or the seed pods, or both, in a balanced diet, or supplementing usually a protein-poor diet with Moringa. I think more concentrated preparations that are enriched in specific phytochemicals, by and large, do and should require additional clinical evidence. But I think if we approach this rationally and promote and fund high-quality scientific rigorous studies, the acceptance of these products and these indications will come in good time. So again, I meant to put this at the beginning, so the tense is sort of screwed up there. Do we have a few more minutes for me to talk about? So the title of my presentation, in the title of my presentation, I gave Mark Olson as my co-author. Many of you know or know of Mark. Mark's a great guy. He's a character. He and I have a very fine relationship, mostly by Skype. And Mark was one of the very early people to start talking about Moringa and start promoting it. He had a website when he was a graduate student at the University of Missouri, in Missouri Botanical Gardens, that had a huge amount of information on Moringa. And as soon as May's 1996 article got me tuned into Moringa, or shortly thereafter, I found Mark's website and began a correspondence with him. And Mark and I published Spanish, sort of an update of my 2005 review of Moringa. And it's in Spanish language, and it's not widely available, but because it was published by his university in Mexico City, and it just doesn't get around that much. But I'd be happy to send that to you if anybody wants to read about Moringa in Spanish. I'm not very good at reading Spanish, by the way, so I have to trust that Mark translated everything we said properly. So what we'd like to do is just talk a little bit about all of the other species of Moringa. We're concerned about preservation of Moringa germplasm for what I hope are obvious reasons. I find it sort of interesting that if you look at the distribution of, that you look at biodiversity maps or you look at global distribution of endangered plants, you all are right in the middle of a hotspot for endangered biodiversity and loss of speciation. This was a fascinating but highly technical paper that was published very recently. So we're concerned that if we don't do something to save the remaining Moringa species, we may not have a chance to utilize them, and some of them may be as good or better as Moringa oleifera. They're just typically, they grow in, by and large, different places and have different characteristics than oleifera. I think this was already mentioned, I don't think I need to dwell on this, but in many developing countries, as much as 80% of the country's population which rely as derived from traditional plants. I think you mentioned 75%. It's a high number. And so, it's important to keep these plants alive and going and to keep up our knowledge of what they contain and what they can do for us. Moringa is native to, Moringa oleifera is native to a very small region in northwest India and Pakistan. Now, this comes from Mark. He and I have talked about this a lot. This is something I can't defend. I have to defer to my other author who's someplace else. So if you want to pick a fight with me about this, I'm sorry, I will lie down. There are 12 other closely related and genetically very similar species. Many of them are edible, in terms, the third point here is that in terms of domesticated Moringa, that you all at Moringaling and pretty much all Moringa growers worldwide cultivate come from a very, very small subset of the genetic diversity of Moringa. You're all familiar with PKM1 and PKM2 developed in India. I'm not certain of the complete spectrum of diversity represented in what you're growing. But the point here is that we're sampling and we're using a very small sector of the germplasm. It originated a long time ago in northern India. And how it moved around the globe, how it became such a prolific plant in the Philippines, for example, I'm not sure. I've talked to Mark at some length about this. So a lot of this gets lost. Very interestingly, though, Mark Olson and a few others in the world now are able to take modern DNA technology and they're able to determine how closely related specific Moringa accessions or cultivars are to the original germplasm. Part of the problem is that we don't have very much of that original germplasm from northern India, and even Mark has repeated this to me multiple times. He can't even be positive when he goes and gets a plant that appears to be growing in the wild, especially in a heavily populated area like Moringa, whether that plant actually was one of the original stands. So there are a lot of issues, but molecular biology, modern technology, looks like it can help us resolve some of these questions, and therefore see how Moringa oleifera and other Moringa species branched out from the original species, and can get a better handle on the biodiversity within the species. So I mentioned that you know that there are 13 species. They're listed here, and by and large they're the bottle tree morphology, they're the slender tree morphology, and then the more tuberous trees and shrubs. Many of the rest of them, other than oleifera, are native to Northeast Africa, Madagascar, other parts of Africa. I've got some pictures. This is from Mark's website, and by the way, he wanted me to mention to you that his website is migrating. It used to be MOBOT, Missouri Botanical Garden, and he's moving it all to this site now, explorelifeonearth.org, Moringa Home. So he's got pictures and some discussion of where they came from, where they originated. It's a wonderful website, and I'll just highlight that so you can go to it yourselves. On that website, and he's had this posted for many years, he indicates where he has collected all of the moringas that he has in his collection. And he went to some very harrowing places and got into some very difficult situations trying to take plants out of some areas where they didn't want to see them. This was a number of years ago. And so what I'd like to focus on in a minute is what we're trying to do with these plants, many of which are actually still in his hands in one way or another. So the shading just shows you the number of moringa species in each of those areas, each of those countries. Again, this can be read on his website. Just go through a few pictures that Mark has supplied me with to show you examples. Moringa druhardi in southern Madagascar. The bark of this tree is used medicinally, and Manny and I were talking about this. As far as I'm aware, most of the moringas have been referenced by traditional healers in their country of origin to have some medicinal use. Moringa stenopetala. Manny showed a picture of a big one. This is, I'm not sure where this one was actually taken. This is in a greenhouse. Moringa ovalifolia from Namibia, very drought resistant, a lot of water storage in the trunk. This is one of the so-called bottle tree varieties. Moringa concanensis, from India, Pakistan, and Bangladesh, is thought to be the closest relative of Moringa oleifera. Moringa peregrina throughout the Red Sea region. It's got edible tubers. Its oil has been prized since for a long time, since antiquity. And actually, David has a wonderful specimen corrected in your greenhouse, and maybe we can talk about that in a question and answer. And I'm sorry, David, you have 4 species of moringa growing now in your farm.
We have Moringa peregrina, we just mentioned. We have Moringa stenopetala. We have from Madagascar, Moringa hildebranti. And of course we have Oleifera. So one of you who wants to see them, they invited to our farm to see them.
Ruspoliana, an example of another tuberous moringa, originally found in a number of places, but now, according to Mark, only in Kenya and southern Ethiopia, and in your greenhouse, right? You don't have that. Moringa borziana, a very small species, but with tender leaves and very large fruits. Moringa pygmaea, also a small species. Moringa ribe, another tuberous tree, Moringa longituba, again from East Africa. This is very small. Usually the tuber in this plant grows beneath the ground because it's been in cultivation in a greenhouse. It looks a little bit different morphologically. So what Mark and I have been working on recently now is a Moringa rescue and living repository effort. And what we've done is we have found with financial support very recently from Trees for Life International, my good friend Balbir Mathur and his organization in Kansas in the U.S. We wanted to take Mark's collection, which, as I showed you, he collected all over East Africa primarily, all over the world really. We wanted to bring those living specimens back someplace where they were under better control. He until a few weeks or a few months ago, had them in Missouri Botanical Gardens, some of them since 1987. And you saw this poor specimen that's flat bound. So he had his specimens being taken care of, watered maybe once every 6 months or maybe once every month if he was lucky, by the caretaker at the greenhouse there. But he moved on to Mexico City to his job. Some of Mark's collection, and again, this is I think the best collection of living Moringa in the world, some of which cannot be gotten again. Some of those plants were at his parents' house in California. They built a little greenhouse structure to house them, and some of them he had in Mexico with him. So the idea, the first part of our preservation idea was get a site and preserve them. And so we've now got, Mark and his wife have purchased a small amount of land in Jalisco province in Mexico. They have taken all of these moringas that have been collected, and now they have them under irrigation, under care. The idea is we want to get them healthy, replicate them. Some samples are coming to my lab to do tissue culture so we can make sure they stay alive. And then we want to replicate this collection to preferably 2 or 3 other places in the world. It doesn't have to be in Mexico or the U.S. It could be in the Philippines. It could be down the road. But we want there not to be the opportunity to lose these valuable specimens. Of course, that takes money and health and willpower. But that's the idea. This is just an example of the state of some of his plants as of a few months ago. They're much better now, because many of them are leaving. But this is a lot of material all over the world. Go through these quickly. He's of course got a seed collection from all of these, and please don't all write or email me or Mark and ask if you can have some of those seeds. The answer is no on most of them, I'm sure, for the time being. But the objective is that we want to be able to distribute these seeds, obviously, but we have to get the plants in better conditions first. Just some more pretty or not so pretty pictures. So I think these are the last 2 slides that I have then. We need to break for lunch and so on. Conclusions. Intelligent use of Moringa requires knowledge of the genetic diversity present in the wild as well as in cultivation. I mentioned this earlier. Wild populations of some of these domesticated organisms are invaluable storehouses of genetic diversity, and we may be able to use them to improve the domestic stocks. By that I mean you may be able to crossbreed some of these accessions that are in the collection with whatever you're using now to make them more drought resistant or have higher protein, more digestible protein, et cetera, et cetera. And we do have, I should mention, we have a proposal that we put together, maybe on the next slide, in which we step by step go through how we want to deal with this germplasm after it's preserved. And I mentioned that the status of the wild populations is unclear, but we're aiming to sort that out by molecular techniques. So the project that we've outlined, we've given it to some philanthropic, potential philanthropic donors. Nobody has funded it. We've given it to the New York Botanical Garden and the Missouri Botanical Gardens. They both want to help, but they haven't funded it. And what we're anticipating is first to produce global maps of the distribution of Moringa oleifera, areas of cultivation, a detailed genetic analysis, enhanced germplasm collections, as I mentioned, and then genetic work on selected attributes, including the nutritional quality and medicinal properties. And this is now possible within the last 5 or 8 years. Science has evolved to the point where we can do some of this. Ultimately, this should result in long-term improvement of Moringa versus via breeding and to clinical trials that establish firmly, by rigorous biomedical standards, its efficacy in various situations. We'd like to acknowledge help from a variety of foundations and funding sources. The last 3 are the March work, the first ones are mine, and some of our collaborators. I won't name them. I'd like to close and again thank you for bearing with me. It was a long talk, but I came a long way. I didn't want to cheat you and give